Greetings, class, and welcome to Chapter 18, Classification. This is going to be a pretty short unit because really the rest of the textbook that we work through in the final quarter of the semester, as we look at the different kingdoms of life, is going to involve talking a lot about classification as well as also talking a lot about evolution again, like we talked about in the previous unit. So we're just going to kind of introduce classification in this particular chapter and talk about how modern classification works and how scientists classify organisms. First of all, why do scientists classify? Well, when you think about it here, there are approximately, just a guesstimate, there's a wide range if you look up at a line, between 8 and 10 million different species that are known on the planet. And that's going to be a small fraction of what actually is out there that hasn't even been identified yet. So imagine trying to make sense of just millions and millions of things. It would be impossible if there wasn't some type of organizational system to do that. And not only that, new organisms are being identified every single day. So they have to be added to this system. So there has to be some kind of method for updating and adding new life as it's discovered. Now, what is classification? Classification is the arrangement of organisms into orderly groups based on their similarities. Like, for example, in your kitchen, I bet you have a drawer where you put your utensils. I bet you have a cabinet where you put your glasses, a cabinet where you put your plates, a pantry where you put your food, and then you probably have your cans on one shelf, pastas on another shelf, whatever. It's human nature to organize things. You have to, because we have so much stuff. Imagine how your kitchen would be if you had your utensils with your glasses. I mean, it would be very difficult to find things. And scientists do the same thing. They classify organisms based on similar characteristics so that it's easier to find them and fit them together and add new organisms. It makes sense and see relationships. This process of doing this is known as taxonomy. It's also known as systematics. That's the more modern, cooler term, systematics. And a person who does this for a living is called a taxonomist. Not to be confused with a taxidermist, but a taxonomist. In fact, this root word, taxon, will be coming back in a couple of slides, and so don't forget it. We'll be using it again. Now, why do scientists classify things? First of all, it allows for accuracy and uniformity in naming organisms. It prevents misnomers or mistakes in naming, and it creates a uniformly recognized language, which scientists around the world share. For example, Greek or Latin derivations for the scientific name, although a common name might be different. The scientific name is what's recognized by the international community of scientists, whereas a common name or a colloquial name for a local area might be different from place to place. Like, here's a great example here. Look at all these different scientists all naming the skunk differently. But regardless of what they call it, the scientific name is the same. So classifying organisms and coming up with a common naming system allows scientists around the world to be able to see how organisms are related to each other, identify organisms in a way where there's no duplication, no overlapping of names, that kind of stuff. Now, taxonomy is not something that just popped up a few years ago. Taxonomy has been around for a long time. One of the earliest taxonomists was Aristotle. I'm sure you've heard of him before. And he had a very early system that divided life basically into plants and animals, which you can see for the little bit of knowledge people had back then about science. That would make sense. Like two kingdoms, plants and animals. And then he divided the animal kingdom into, for example, organisms that dwelled in the land, sea, or air. But I bet you're saying to yourself right now, I can see a problem already. There are organisms that live in all three areas. Where would they go? So that was a recognized system for a long period of time. But then later scientists tried to revise the system to make it more efficient. Another scientist who's got some hand in the contributing to the development of modern naming system is John Ray. He's one of the first scientists credited with using Latin for naming. Problem was, in this period of time, using Latin was nice, but they didn't really have a defining way to come up with the names. So the names are very long, too descriptive, and very, very complex. So scientists recognized they needed a better system for naming, something that was simple, efficient, and could be universally recognized and accepted by scientists. So Carolus Linnaeus was an 18th century botanist and taxonomist, and he developed the modern classification system that we use today. In fact, his system is internationally recognized. And although it has evolved, the basic core of it is still what scientists use today. And it actually fits in perfectly with what we talked about in the last unit. There's a tremendous relationship between Darwin's work and Linnaeus's work. And if you remember in the Bill Nye video we watched, in fact, modern biology really is evolutionary biology. And these two scientists' work are intricately intertwined with each other, even though Linnaeus came before Darwin. It didn't have the wisdom of knowledge in terms of natural selection and evolution. That will come much later. But Darwin's work fits in perfectly with what Linnaeus did. Now, Linnaeus is considered the father of modern taxonomy, just as Mendel is considered the father of genetics. He developed a system that we call binomial nomenclature, which means two-part name. You can see the prefix there for two, nomial naming. And his two-part naming system consists of the genus and the species, which are levels of classification we'll get to in a second. The rules are very simple. The name is given in Latin or Greek. It's italicized. You capitalize the genus, and the species is left lowercase. So you have the genus right here, and then the species right here. Here's some examples here. You can see 3 different bears: the panda bear, the polar bear, grizzly bear. Which two are most closely related? Look at the genus. Grizzly bear, polar bear, both share the same genus, so they actually go down the most levels of classification and are the most similarly related. These are the levels of classification, starting with the domain. The domain is the broadest taxon. That is the most recent addition. In fact, when I was in school, we didn't even learn domain. That's 35 years ago. The domain has been added recently to recognize some changes in knowledge that scientists have. Linnaeus' system started here, so be careful when you're answering questions. The Linnaean system started with kingdom. Our modern system has the domain. So currently today there are 8 levels of classification. Broadest, of course, means the biggest, and then the word taxon, which you probably saw that root word a few minutes ago. A taxon is an individual level of classification in the system. And then as you go down, it's kind of like a reverse pyramid. Every level has less and less organisms until you get to the species. At the species level, you have one organism. Those are organisms that are very similar, can mate with each other, reproduce, and produce offspring that can also reproduce. Now, how do scientists come up with a naming convention for organisms? When a scientist discovers a new organism and decides to try to give it a name, that name would have to be accepted and recognized by the international scientific community. That's very important because we don't want organisms with duplicate names or scientists naming an organism that already exists that somebody else knows about. So scientists will have to meet and discuss these organisms when they're discovered and come up with consensus for a name. Remember we mentioned a second ago that word taxon or taxa, which is the plural. That's the category or level of classification an organism is placed in. It is a hierarchy from broadest to most specific, like I said, and these are the levels of classification in the modern system today. Looks like this. Here's a terrible mnemonic that you can mnemonic device you can use to remember. Dumb King Philip came over for a gooseberry soup. But you can see each letter there. The red ones highlight the actual level of classification. This particular slide here doesn't show the domain, but it does start at the kingdom, and you can see every level it gets less and less organisms. Like the Animalia kingdom, you can see there's quite a wide variety of organisms in there. But as you go down each level, you see things disappear. Like first the starfish disappears, then the snake disappears, and so on and so forth until you get to the bottom, which reflects back to the slide we had a few minutes ago with the grizzly bear and then the polar bear. And look how closely these guys are related all the way down to genus level. The domain is the broadest, most inclusive taxon, which means it has the most organisms in it. There are 3 domains today. There are archaea and bacteria. These are 2 different domains. They're unicellular prokaryotic organisms, but they're placed in their own domains because scientists have found recently there's just so much difference between archaea and bacteria that they really need to be classified separately. And then the third group, the one we belong to, is Eukarya. This group differs from the other two in that these guys are eukaryotic and have a nucleus. Once you go through and divide an organism into the appropriate domain, you could have a domain with millions of organisms in it. So then those organisms have to be reclassified again. So then you start to go through the levels. In this particular chapter, we're just going to highlight the kingdoms, and then as we go through the rest of the book, we'll look at the different levels of classification as we get more specific and move into the animal kingdom, the plant kingdom, that kind of stuff, and then go down phylum and work our way down, looking very specific at some of the organisms representative of each of those groups. First kingdom we'll look at is Archaebacteria. Remember the word archa, scientific root word there, means ancient. These are some of the organisms that are oldest living on our planet. These guys have basically been around since the beginning of time. They can live in the most inhospitable of environments. They can live in areas where there's no oxygen. Those guys are called methanogens. They can live in areas of extreme heat that would kill modern-day bacteria, the thermophiles. They can live in salty areas, which would kill most organisms, the halophiles. So these are a very hardy group of bacteria. This is one of the reasons why they've been separated from the modern bacteria. The two groups together once upon a time used to exist. Here's an example of some Archaebacteria. Used to exist together in a group called the monorans, but have since been separated because now that with modern science, the molecular structures, the cell walls, there's just so much difference between them, they couldn't be classified together in the same kingdom anymore. Your Eubacteria, or your modern day bacteria, and so there's actually some dispute over this name, which they may be changing in the near future as well. These are your modern day bacteria. Some cause disease, some are consequential to us, some are also very beneficial. In fact, most bacteria we have no consequence with or really are helpful to us, and life wouldn't be possible on Earth without them. You find them everywhere. You've probably got some around you right now. They're important decomposers, recycling nutrients into the soil. They're very important commercially as well. You have them in your body helping you break down your food right now. So these guys are very important. They can't live in the same types of harsh environments that the Archaebacteria do. So consequently, they're in their own kingdom. Here's some E. coli right now. In this picture here, those are in your intestines right now, how you break down your food. Third domain is Eukarya. These guys here all have a nucleus. Wide-ranging domain here. You have Protista, fungi, plantae, and animalia. When you're studying for the quiz, make sure that you know the characteristics of each one of these groups. Like, for example, the protists, this is what they call the junk drawer kingdom, because this is where scientists put organisms where they have no idea where else to put them. But they do share some things in common, like they're multicellular. They're unicellular. But although some, like algae, are multicellular, the vast majority are unicellular, and they live in aquatic environments. But you have some that are autotrophic, some that are heterotrophic, some are fungus-like, some are plant-like. So there is some discussions that one day this might be the next kingdom that's broken up. Protista might be broken up into groups one day as well. But for today, it is its own kingdom. So if something is an animal-like organism, plant-like, or fungus-like, and it's unicellular and likes to live in water, they'll throw it in the protist kingdom. Then you have fungi. Fungi are multicellular, except for yeast. They're absorptive heterotrophs, which means they actually digest their food on the outside and then suck it up through these tube-like structures called hypha. They have cell walls of chitin. In fact, once upon a time, fungi were classified with plants, and you could probably understand by looking at them why. They're non-motile, like plants. They kind of just sit there, and they almost even seem to have a root system, although it's not a true root system like in a plant. But these guys are heterotrophs, and their cell walls are completely different from plant cells. So today, they're in their own kingdom. Plant kingdom, these organisms are multicellular, autotrophic. Remember, they undergo photosynthesis. We talked about that earlier in the year. And they have cell walls made of cellulose. And finally, the kingdom Animalia, to which we belong. These are multicellular organisms as well. These guys are, like us, are ingestive heterotrophs in which we consume our food and then digest it in our bodies. And we feed on plants and or animals. And this is the last group we'll look at in the semester. And in fact, the rest of our units will pretty much follow in order the way I just laid them out in the presentation here for you. And this is a great chart to make sure you look over for the quiz coming up so that you know the characteristics of each group, as well as the chart you made in class the other day. Now, let's move really quickly into how scientists classify things. early classification systems were based more on structure of the organism, physiology, that kind of stuff. But now scientists look more into things like evolutionary relationships. They look at some of the stuff we talked about at the end of chapter 16. A lot of that evidence that we talked about for Darwin's natural selection are some of the pieces of evidence that scientists use to classify organisms. They look at things like homologous structures. They look at embryo development. They look at the microbiology of the organisms and the molecular biology. These kind of things are used to help organisms today. And the more levels of classification organisms share with each other, the more similar their evolutionary histories are. Here's some examples of things that scientists look at. Homologous structures, you remember these. Embryo development, you remember that one. Now, these are some new tools that we're going to talk about here. First is the cladogram. This is a tool that taxonomists use to show the evolutionary relationship between organisms. They're very easy to use. The cladogram usually begins with a common ancestor at the beginning. And what it shows is the evolutionary flow of the organisms as they develop and the specific adaptations or characteristics that develop amongst these lines of organisms as they evolve and develop. The more characteristics organisms share in common with each other, the more closely related are. These characteristics here are called derived characters. And these mark significant evolutionary developments when these particular structures appear. An organism to the left of a derived character does not have the character. An organism to the right of the derived character does. Where an organism breaks off from the common line of the cladogram is called a node. This is where they differentiate. So, for example, you can see the lancelet here. The node here shows that the lancelet does not have a vertebral column. The vertebral column appears after the development of the lancelet. But you can see all the other organisms do. The lamprey does. The grouper does, the salamander does, the turtle does, and the wolf does. How about jawbones? Do lampreys have jawbones? No, they do not. The jawbone appears after the lamprey develops. You see its node appears before the jawbone. Lancelet does not have a jawbone either. But the first line that we see here in the cladogram that does appear to have a jawbone are the grouper. And then you can just go down the line here. You can see the differentiating characteristic that separates the wolf from the rest is the appearance of the hair. And so the cladogram is a very good tool to use to see relationships between organisms, how closely related organisms are. You can see, for example, the distance between the wolf and the lancelet. You can tell that they do not share quite a similar evolutionary history at all. They're very far apart and quite dissimilar from each other, although they are both chordates, but the rest of their evolutionary history is pretty different. You can see there are many more derived characters shared by, like, for example, the turtle and the wolf than the wolf and the lancelet. So that's one tool that scientists use. Here's another example. This is a primate cladogram, which you can use to review as well, and we practice this in class. You can see again the appearance of the derived characters, and then, of course, the nodes where the organisms shoot off. Another tool that taxonomists use when dealing with organisms and identifying them is the dichotomous key. The dichotomous key, the prefix di, means two, and basically what it is sets of paired questions that scientists can ask to help identify an organism. Each paired question is a set of characteristics, and each set of questions will direct you to the next set of questions or will help you to identify the organism. Very simple tool, but very powerful. Here's a great example. Let's try to identify this organism right here in the bottom. Does it have tentacles? That's the first question. You see 1A, tentacles present, or 1B, tentacles absent. So that's the characteristic that's being asked in the two questions. If it has tentacles, you go on to question 2. If there are no tentacles, you go on to question 3. And in this case, it does appear to have tentacles, so I'll go on to question 2. Does it have 8 tentacles? If it does, it would be this guy here. But it does not. It appears to have way more than 8. So I'm going to go on to question 3. Do the tentacles hang down? If they do, then I would go on to question 4. But they appear to be upright. And with answering those 3 questions, I'm able to identify this organism as a sea anemone. So, that concludes our study of classification in terms of the introduction. The rest of the units that we'll be studying into the last marking period, we're going to continue our work with classification and evolution, tying it all together as we look at the different kingdoms of life. Don't forget to look over the presentation. Of course, review this video, which you have. Look over all 3 readings. Do the practice questions at the end of each section, and you'll be ready for the quiz on Tuesday.